students. Today we're going to discuss about respiration, which has three major processes responsible for yielding energy in the form of ATP, which is adenosine triphosphate. Okay, so first, respiration is a process in which glucose molecule is being converted into energy by using oxygen as the primary tool. So the first step which I am going to support you in today will be glycolysis. So in glycolysis, first you have to know and you have to discuss that it requires oxygen. It occurs where? So you have to learn the location. So we have talked about a different overview about glycolysis, and moving on to the diagram here, I'm just going to explain it to you from the diagram as well. Just wait a second. Yeah, so here the aerobic respiration, which is glycolysis, which is a part of aerobic respiration, because in anaerobic respiration is fermentation, and I have explained that to you as well. So glycolysis is the first step of ATP generation. It is the first step. Second step is the citric acid cycle, or also termed as Krebs cycle by the scientist Sir Hans Krebs. And the last stage for generating ATP is oxidative phosphorylation. So glucose is converted into pyruvate, and pyruvate is then being driven out and transferred to the mitochondria. So all the process which is happening here of glycolysis is obviously being carried out in the cytosol, which is the liquid part of the cytoplasm or the fluid part of the cytoplasm. So basically, cytosol is that part of the cytoplasm which is fluid, which has a less nutrient content, and it is opposite and posterior to the plasma membrane. So obviously, after all this process happening in the cytosol, the pyruvate enters the mitochondria where the citric acid cycle is basically happening. So what is glycolysis and why does it occur and what happens there? We are going to discuss it here. The definition of glycolysis is that it is the first common step for aerobic and anaerobic respiration, and glycol means glucose, lysis means splitting. So the splitting of glucose in the presence of oxygen will lead to pyruvate, and it will generally lead to the ATP production. So basically it is a common step. It is the first step for both aerobic and anaerobic respiration. While aerobic requires oxygen in a definite amount, and anaerobic it can prosper without using oxygen in that definite amount, but it occurs mostly in yeast or in certain bacteria. But the aerobic respiration occurs in all the other organisms which are consumers. Moving on to the chapter, you have, I think you also have ecosystem and ecological chapter in your syllabus as well. So it is a part of it that aerobic respiration occurs in consumers and producers, while anaerobic can occur in yeast and bacteria. So okay, so here I was telling you about that C6H12O6 is glucose, while C3H4O3 is pyruvate. And moving on to the further explanation of it, it is defined as a metabolic process where a glucose molecule gets broken down under the influence of several enzymes. So it is very important that we have certain enzymes as well, and we have to learn the names of them as well. The 6-carbon compound glucose, which has 6 carbon, and it splits into 2 molecules. It means that it is going to split into 2 molecules of pyruvate, and obviously it will also yield ATP. It will yield ATP, NADH and water. It will yield ATP, NADH and water. What is NADH and how is water released? We're going to discuss the first phase of glycolysis, and we're going to discuss the cycle of glycolysis. So the first phase of glycolysis is mentioned here. I've mentioned here for you guys so you can have a proper idea and a proper description of what is happening. So in the first step, a phosphate group from ATP gets transferred to the glucose molecule we are present in cytoplasm, producing glucose 6-phosphate molecule, and this step is catalyzed by enzyme hexokinase. That means that this process occurs in the presence of this enzyme. Okay, so in the reaction, glucose is converted into glucose 6-phosphate by hexokinase and taking a phosphate molecule from ATP. So basically, C6H12O6 is given a phosphate group from ATP. So here energy is being provided. Energy is provided. I've mentioned here as well that the overall process, it is converted into two phases: energy requirement or energy investment. So that means that here energy is invested by ATP using a phosphate from ATP, converting it into ADP, as ATP has 3 phosphate groups. One is released, two glucose, then 2 phosphate groups to ATP are left, then it is converted into ADP, triphosphate, diphosphate 2. So now, because glucose is being given a phosphate group from ATP, that's why it is called glucose 6-phosphate, because with the 6 carbon of glucose, a phosphate group is attached. That's why with the carbon number 6 of glucose, another phosphate group is attached. That's why it is called as glucose 6-phosphate. Then in the next step, in the glucose 6-phosphate gets converted into its isomer fructose 6-phosphate by an action of phosphoglucose isomerase. So this enzyme is generally responsible for the isomerization of glucose. So let's name these reactions. The first one will be phosphorylation of glucose. Why? Because phosphate is added, and when something is added, you have to add the word TION to it, add the phrase. So basically, phosphorylation, addition of phosphate. If phosphate is removed, then we will say dephosphorylation. Okay. So after this process, when glucose 6-phosphate is converted into fructose 6-phosphate, and you should also know that both of these are isomers of each other. So because they are isomers of each other, because glucose as a whole, let me tell you, glucose is an aldehyde, fructose is a ketonic group. That's why glucose and fructose and aldehyde and ketones are isomers. So glucose 6-phosphate and fructose 6-phosphate are isomers of one another because they have the same phosphate group at the same carbon. That's why glucose 6-phosphate can be converted into fructose 6-phosphate by the presence of enzyme phosphoglucose isomerase. And this process can also be termed as isomerization of glucose 6-phosphate. Then the fructose 6-phosphate, which is isomerized from glucose 6-phosphate, is being converted into fructose 1,6-bisphosphate. Okay, so this is a very interesting term. Let's see what is said here. So another phosphate group gets transferred from ATP to fructose 6-phosphate to produce fructose 1,6-bisphosphate under the influence of phosphofructokinase. So we have the name of the enzyme as well. So fructose 6-phosphate, because it is obviously containing one phosphate, and when another ATP, so till here we can say that 2 ATP molecules are being added, energy is being provided. So this is energy driven process. Energy is given in this process, and this process is done and is taking place by the use of energy derived from ATP. A glucose 6-phosphate transferred to fructose 6-phosphate and fructose 6-phosphate to fructose 1,6 biphosphate, because from ATP now again another molecule of phosphate, another mole of phosphate, inorganic phosphate is taken. It will again convert into ATP and inorganic phosphate. This phosphate goes to fructose 6-phosphate. It converts to fructose 1,6 biphosphate. Now fructose contains two phosphate groups, one at carbon number 1 and another at carbon number 6. That's why it is called fructose 1,6, the position of the phosphate group, bi, because two phosphate groups are present. Now the next step, because till now we have covered 1, 2, 3 steps. So this step will also be called as a phosphorylation, but of fructose 6-phosphate. Okay. Then the next step, moving on to the next step, we are at the 4th step. The enzyme aldolase acts on the fructose 1,6 biphosphate and splits it into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And these 2 3-carbon molecules are isomers of each other. So in this reaction now, this is one of the most important reactions. Aldolase enzyme, it converts fructose 1,6 biphosphate, because fructose 1,6 biphosphate is a very large molecule. It is a big molecule. It has become a polymer even. So now it has to be differentiated into 2 molecules. So because it is 1,6 biphosphate, so it is now broken down into 2 molecules of 3 carbon atoms. One molecule is called as glyceraldehyde 3-phosphate, also known as G3P, and dihydroxyacetone phosphate, DHAP. These 2 are also isomers of one another. So it is very prominent till now that G3P can be converted into dihydroxyacetone phosphate, and this can be converted into each other as well. So in the above reactions, let me tell you this one thing which I think you might have observed. You can observe double arrow here. You can observe double arrow here, here, here. It means that this process is reversible. Fructose 6-phosphate and glucose 6-phosphate are isomers, they are reversible. Fructose 1,6 biphosphate and conversion into 2 molecules is reversible. These 2 can mix and make that again. That molecule can break down and make these 2 again. So just to make it clear to you guys, I wanted to make it very obvious that where double arrows are mentioned and where they are not. Okay, so moving on. When glyceraldehyde 3-phosphate is converted, is made with dihydroxyacetone phosphate, then as both of them are the isomers of one another, it is very obvious that it can also become this and it can also become this. So as I mentioned here as well, so first we had 1 molecule of G3P and 1 molecule of dihydroxyacetone phosphate, and now we have 2 molecules of G3P because dihydroxyacetone phosphate has been isomerized to G3P. Now we have 2 molecules of glyceraldehyde 3-phosphate. So after this, moving on to the next step, in which glyceraldehyde 3-phosphate is being given something. Firstly, it is given NAD, then it is being given phosphate group. Obviously, it is again from ATP. So the basic process which I am going to explain here again which we discussed as well. Let me point out the process and the step which we have to discuss. Yes, this is the step. The enzyme G3P G3P, the enzyme glyceraldehyde 3-phosphate dehydrogenase. So G3P dehydrogenase enzyme is responsible for removing hydrogen from G3P, and it adds a phosphate from the cytosol to the glyceraldehyde 3-phosphate to form 1,3-bisphosphoglycerate. Okay, so again it is interesting. Let's see. I have to explain this to you here. We had G3P. We have two molecules of G3P. Okay, so from cytosol, where the glycolysis process is occurring, two phosphate molecules are being given to G3P, and along with an enzyme acts on G3P, which is G3P dehydrogenase enzyme, and this dehydrogenase enzyme is responsible for removing hydrogen from it and giving phosphate ions to it. That is why here in this process we can say that 2-NAD in the cytosol by the removal of hydrogen from G3P become 2-NADH. That's why NAD is converted into NADH, and this phosphate is not from ATP, because I was tricking you guys, it is from the cytosol. Because you can't see ATP here. If ATP was written here, it would have written ADP or inorganic phosphate, or the ATP would have been written here. But this is the process where it starts from this process, from this area. The energy producing or energy yielding process starts. Okay, so because there were two molecules of G3P and both of them released their hydrogen ions and accepted phosphate, and because they accepted two phosphate ions, that's why they will have phosphate at two positions. And because it is written 1,3-bisphosphoglycerate, it means one phosphate group is at carbon number 1 and one it is at carbon number 3. So after that, 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate by an enzyme called phosphoglycerate kinase, is responsible for converting 1,3-bisphosphoglycerate to 3-phosphoglycerate. What is happening here? So we can see that from carbon number 1 position, it is now only 3 position is visible, because the phosphated carbon number 1 is being released and it is used by ADP in the first phase to form ATP again. So now the ADP, which were deprived of energy and their inorganic phosphate, become ATP again. So now we have two molecules of ATP here firstly, then this bisphosphoglycerate molecule 1,3 only becomes 3. Now it has just one phosphate group at just carbon number 3. Then this 3-phosphoglycerate is being converted into 2-phosphoglycerate by what? By phosphoglycol mutase enzyme, and that is responsible for the isomerization. So again, I'm going to point out it for you so that you have a clarity about it, what I'm talking about. You can see here that 3-phosphoglycerate is converted into its isomer to phosphoglycerate by the action of phosphoglyceromutase enzyme. So here as well it is very clear that both of them were isomers, and we can also say the isomerization of 3-phosphoglycerate is 2-phosphoglycerate, and obviously that will lead to 2-phosphoglycerate from 3-phosphoglycerate. From third carbon, phosphate from third carbon is transferred to second carbon by this enzyme. Now this 2-phosphoglycerate is converted into phosphoenol pyruvate molecule by the enzyme called enolase. So you can see that enzyme is enolase, enol. So enolase enzyme for converting phosphoglycerate to phosphoenol glycerate to pyruvate just by the removal of 2 moles of water. Enolase enzyme converts 2-phosphoglycerate to phosphoenol pyruvate, and now by the work of the last enzyme pyruvate kinase, phosphoenol pyruvate is converted into pyruvate just by the process of the removal of phosphate again, because pyruvate does not contain any phosphate. The formula is C3H4O3, no phosphate ions. So only phosphate is being released by here, phospho group is released by phosphoenol pyruvate, and only pyruvate is left, and these phosphate ions released are also taken by the ADP used in the first phase, and then we have 2 molecules of ATP. So we have discussed all the steps of glycolysis from first step towards the last step, and moving on to the equation, you should know that the reactants we had in the first phase were glucose, NAD, ADP, adenosine diphosphate, and inorganic phosphate, which was used by here, by glucose 6-phosphate, by fructose 1,6 bisphosphate here as well. So phosphate that was used in the first phase was given out in the second phase just for the production of ATP and pyruvate, which will give us more ATP in the next step. Products were pyruvate, NADH, which was given here by 1,3-bisphosphoglycerate, and G3P, which already had 3-phosphate. Hydrogen ions were given, which were taken up by NAD, forming NADH, nicotinamide, adenine dinucleotide, and water was also formed here. So first phase was energy investment. We used ATPs to drive the reaction, and the second phase was energy payoff. We required the molecules and we required energy for them to give us, because the energy was being provided prior to it by ATP to them as inorganic phosphate. In the end, we had 2 NADH molecules. We had here 2 NADH molecules, and we had 2 ATPs. One was generated here and one was generated here, and we had water as well, 2 moles of water, 4 molecules of ATP, and 2 molecules of NADH. So the reactants are clear, the products are clear, and we're clear about glycolysis. So glycolysis is the first step, it is the first part of generative respiration, and I think we have cleared it till now. And tell me if you have any questions regarding it, any MCQs, any questions. I am free to tell you about it, and I hope it is very clear to you, and you can grab it and move to Krebs cycle in the next process and the next lecture. Till then, take care. Allah Hafiz.